All right, welcome back, ladies and gentlemen. It's been a little time since I've done a recording and an upload, and today we're going to be looking at cell cycle and mitosis, and we're really going to be looking at the important things to note, particularly for the Unit 1 curriculum and the Year 11 curriculum, I should say, and what we need to understand for moving forward in this knowledge point. So, it's no secret that we grow as human beings. We start off as tiny little infants, and then we grow up into adults. And along with this growth, this is where our important notes for the cell cycle, and particularly mitosis, comes into it. Because in order for us to grow, we need to gain more of these. These being the little cell that we got from one of those past diagrams, and we start off with one of them. And then that one turns into 2. And then those 2 turn into 4, and those 4 turn into 8, and so on and so forth. This comes from one of those really important notes from back when you're probably looking at the cell theory, that all cells come from pre-existing cells. We all started out as one cell, and then that cell divided to multiply. It divided to multiply. It went through this process that we're going to talk about today, the process of cell division. All right, so we divide, our cells go through a divide to multiply in biology. So let's talk about this. And we're going to start talking about the interphase as in the cell cycle. In order for this to be spoken about clearly and for us to gain a kind of just a basic understanding, we're just going to refer to a lot of what is happening in the nucleus and the cell body, so the cytosol, or the amount of cell we have. We're going to ignore the rest of the organelles and stuff, except for a couple mentions as to what happens to them in each stage. So we're starting off in interphase. Interphase will start off here at this arrow, and we're entering the G1 stage of interphase. During this stage, the cell goes through an exponential amount of growth. G1, gap 1, growth stage 1, however you want to word it, it's all the same thing. So preparing to divide by growing, we're still actively going through all of the processes during this stage. So there's still all of the cellular metabolic processes, cellular respiration, photosynthesis in plant cells, packaging, creation of proteins, sending them around, all that stuff is still happening. During this stage, the organelles also duplicate in preparation for division as well. So they're making new organelle groups and organelle materials for the new cells. This process, takes around about that 8 to 10 hours. So it's a long process. Interphase is actually quite a long time that it happens over. And that's all we need to know for growth 1 stage, just that the cell is actively still going through processes, all the organelles duplicate, and then it grows during this stage as well. We also need to know that there is a checkpoint at the end of the first growth stage. Now, we're going to talk about checkpoints in a lot more detail in the next lesson, but we're certainly going to mention that here there is a checkpoint, and that the cell is checked at the end of this stage. Focusing in on the nucleus now. The nucleus doesn't necessarily grow, but I couldn't fit the pictures in nicely in that tiny little diagram, in tiny little version, so I've enlarged the nucleus for what happens next. And the next stage of interphase is the synthesis, or S stage of interphase. As the name suggests, this is where the DNA inside of the nucleus all duplicates. And we end up with enough DNA for us to split our cell in half so that they are exactly identical to each other. So all of the chromosomes inside of your cells are going to replicate. In this example, we've gone from having 3 chromosomes to 6 chromosomes inside of the nucleus, and they're all paired up as such as well. If you're not sure what a chromosome is, don't stress. It's usually covered in Unit 2 biology. If you do know what a chromosome is, thanks to Year 10 or something like that, then perfect. You're already zooming ahead. But for now, all you really need to know is that a chromosome is packaged DNA. The other thing to note is that you wouldn't be able to see chromosomes like this in this stage of mitosis. I've just drawn them like this or put them as diagrams like this for representation purposes. They don't actually package up like this until a later stage, which we're going to talk about. All right, moving on from S stage, we get to the G2 stage. As the name probably suggests, it's the second gap, gap 2, the second growth stage. It's all the same. G2 comes after S though. During this stage, the cell continues to grow. All those organelles are packaged up to get ready for the division of the cell. And at the end of this stage, there is another check. A check of the DNA to make sure that it's replicated properly, the check to make sure the cell has in fact grown enough, to make sure that all the organelles are packaged up and things like that. So the cell really focuses in on making sure that it is ready to divide before getting to the final stage of the cell cycle. Now, the final stage of the cell cycle is M phase. It's the mitotic phase. It's where we are now going to get the cell ready to go through nuclear division, where the nucleus splits apart, and then finally finish off with cellular division, where the cell then splits apart. And that is going to be happening all in the, where are you? There you are. All in this top phase up here. I'm going to go through it in a decent amount of detail, let you guys know the important things that you need to know for these stages, and then we'll finish off after that. First stage of mitosis is prophase. PR for prophase, PR for preparing to divide. Another thing to note, for each of these stages, there are also lots of resources out there talking about late prophase, early prophase, pro-metaphase, early anaphase, late anaphase, and things like that we could talk about in this video, but I really want to try and keep this one under the 20 minutes. So I'm only going to focus in on the big four, followed by the last one. So during prophase, spindle fibers appear along with centrioles, and the nuclear envelope disappears. So the nucleus, or the nuclear membrane, that's referred to as the nuclear envelope, dissolves, disappears, and these centrioles appear. So centrioles in during the rest of the stage, these are your centrioles. They're going to be about most of the time in an animal cell, and coming off of those that you're going to see in a minute are these spindle fibers. They are protein projections that come off the centrioles, and they're going to play a very key role in the mitotic process, and they need to form during this process, during these stages, to make sure that the process actually works. So yeah, we're going to be looking out for the spindle fibers. We're going to be looking out for the centrioles during the process of mitosis, and we're going to see how they perform. Right, so couldn't get the pen to work before. Here we go. Now it's working, and we'll move on from here. So that's prophase. Prophase done. Spindle fibers, nuclear envelope dissolves. Spindle fibers appear. Centrioles appear. Time to move on to metaphase. Metaphase. MET for met. At the beginning of metaphase, and also MET for met in the middle. Key thing that happens during this process is that the chromosomes all line up in the equator of the cell in pairs. The other thing you'll notice is that the centrioles move to opposite poles of the cell. So if you can imagine a dotted line going all the way down the center of the cell like that, that would be the equator, much like the equator of a planet, and these guys then move to the north and south poles away from that equator. What also happens is the spindle fibers then move and attach to the chromosomes at where the X would appear I've forgotten the word. Sorry, where the X appears in the chromosome structure. And this is what these are what we call centromeres. This is the attachment point during metaphase. Another thing to note, actually, that is slightly important, I'm going to go back a couple slides, is during prophase, that is when the chromosomes condense and they become visible. I forgot to write that down on my notes. It's probably important for you guys to write that down, that during prophase, that is when we can actually see chromosomes like this. All right, move forward again. There we go. Metaphase, we've met in the middle and we've attached. That's the important thing. We line up our chromosomes in pairs at the equator and they all attach like this. There's also a check at the end of metaphase. So that's 3 checks in total. Three checks in total. We have had one at the end of G1, one at the end of G2, and one at the end of metaphase before moving on to the next stage, anaphase. The major purpose for this one is to make sure that all the chromosomes are in fact lined up in the equator and that all of the spindle fibers have indeed attached. It's important because if spindle fibers don't attach, then the next stage will not happen properly. Anaphase. A for anaphase, A for apart. And just like this next one states, this is a stage where the spindle fibers are going to pull sister chromosomes to opposite poles of the cell. These guys are chromosome pairs, homologous chromosomes, and as you can see, over they go. It's also key to note that towards the end of this stage, the cell will elongate, so it'll kind of look a little bit more oval in shape than circular if it's an animal cell, and the purpose for this is that it's now getting ready to divide. If those spindle fibers weren't attached properly, then the chromosome pair would all get pulled to the one side of the cell, and so you would have an uneven distribution of chromosomes on either side of the cell for mitosis. The last stage is telophase. I don't have a good word for telophase that it's like. Telophase is where the spindle fibers and centrioles will dissolve and the nuclear envelope will now appear like that. And just like the name, just like it says down here, we're now done with nuclear division. We're done with splitting the nucleus. Mitosis as we know it, or should know it, is now complete. Cell division is not. And that's where our final stage comes into it, cytokinesis. Cyto meaning cytosol, kinesis being moving. We're now going to move the membranes apart. And it's different in plant and animal cells. When an animal cell goes ahead and starts to pinch in the center, what we call a cleavage furrow appears in between the two cells. It's a pinching section where the membrane starts to try to reform and join and touch each other in either sides of the cell. So as it's kind of moving in and the pinching occurs, this cleavage furrow will form in the center, getting ready for the cytosol, the new membranes to join together. And once that happens, the cells can move apart and live happily. One of these cells can go off at the end of this process and go into what we call G0 phase or G0 stage of mitosis, where it goes through its regular functions. It is no longer going to replicate. One of the cells goes back into the cell cycle and goes back into G1 stage, goes through the growth and functions and synthesis, and then growth 2, and then PMATC, and then, same going. Now, if there's rapid growth happening, like when you're an infant growing into an adult, you may in fact have both cells just continuously replicate. But we're going to talk about why that could be a bad thing in next lesson. I did mention that it is different for animals versus plants. This is the animal cell example with a cleavage furrow. Plant cells, up until this point, cytokinesis, are exactly the same as animal cells. The only difference is now when we're splitting apart. A lot of plant cells act like building blocks. They just keep building and building and building upon each other. They don't actually split and then drift away from each other. They just keep building and building and building and building and building. So what you end up with is this example, or this situation where all we want to do is build a new membrane and a new cell wall in between us. During this stage, all the organelles are now back and functional inside the cell. The organelles responsible for creating cellulose in the cell, which is the product that the cell wall is made up of, will send these little bubbles of cell membrane called vesicles towards this equator line here in the center. These little bubbles are packages filled with cellulose. And as that cellulose all meets in the middle and the new membrane starts to form, you end up with this cell plate that forms in the center. It is an incomplete cell membrane. It will just continuously build upon and build upon and build upon. Sorry, not cell membrane, cell wall. You just build upon and build upon and build upon this new cell wall using all this new product until finally you do in fact have a brand new cell wall separating two brand new cells. Eukaryotic cell cycle is, complicated. There's a few different steps involved and things like that, but it's also really important for our curriculum to know what prokaryotes do. So bacteria divide in a much simpler way. It's called binary fission. We're only going to focus again on the cell. Here's the bacterial cell wall, and there is the bacterial DNA, the plasmid, that circular DNA. As it so happens, it is really easy, it's really easy for that bacterial DNA to replicate. It replicates itself and it splits to either side of the cell. In this process, the cell can then split into brand new, taking that DNA with it. I know the animation was a bit janky there, but this one works proper, I promise. So again, it just replicates and then splits apart. See, a little bit better. So we end up with this, lovely situation where we have enough DNA to spread around. The DNA is able to, replicate at a really, really fast rate, and that is how our bacterial cells manage to keep themselves, going and spreading colonies. And this process doesn't take, hours for the mitosis to actually go. For the interphase to go through, and then mitosis to happen over a few seconds. It happens very quickly. We're talking this whole process. You can end up having a split once every 20 seconds in some species of bacteria. Okay, that'll do it for this lesson. Managed to keep it under 20 minutes. Pretty happy with that, and hopefully the edit turns out just fine. Thank you. Catch you on the next one.